Vaccines, in short, are just a way of tricking your immune system to think it is fighting a disease, but in reality, is not the actual disease. Weakened or harmless forms of a disease pathogen, virus, or bacteria is introduced into the body. Once the immune system detects it, white blood cells will be deployed to fight it, and because it is weakened, the immune system will easily eliminate it, but afterwards develop resistance to it. They are meant to make the immune system produce antibodies against the actual disease. Vaccines are a critical tool in public health, designed to protect against infectious diseases by stimulating the body's immune system to recognize and fight off pathogens. The concept of vaccines dates back to ancient practices in China and India, where people would expose themselves to small doses of pathogens to build immunity over time. However, the modern history of vaccination began in the late 18th century. In 1796, Edward Jenner, an English physician, made a groundbreaking discovery that laid the foundation for vaccines. He observed that milkmaids who had contracted cowpox, a mild disease in cows, were immune to the deadly smallpox. Jenner conducted an experiment where he inoculated a boy with material from a cowpox sore and later exposed him to smallpox. The boy did not develop the disease, proving the effectiveness of this method, which Jenner called vaccination, from the Latin word vacca, meaning cow. Later on, there was further advancements in vaccine development. In the 19th century, Louis Pasteur expanded upon Jenner's work. Pasteur developed vaccines for rabies and anthrax through a process called attenuation, where he weakened the pathogens so they wouldn't cause disease but still triggered immunity. His work laid the foundation for modern vaccine development techniques. In the 20th century and beyond, major advancements, with vaccines developed for diseases such as polio, tetanus, measles, mumps, and rubella. Jonas Salk's development of the polio vaccine in the 1950s marked a significant turning point, nearly eradicating the disease worldwide. As technology evolved, scientists also developed the recombinant DNA and messenger RNA vaccines, including the recent COVID-19 vaccines. Vaccines have dramatically reduced the incidence of infectious diseases and saved millions of lives. The success of vaccination programs has led to the eradication of smallpox, declared in 1980, and the near eradication of polio in most parts of the world.
So in reality, you are injected with the same microorganism that is causing the disease or a substance obtained from the pathogen, but don't panic, they can't harm you. What are vaccines actually made of? A vaccine typically consists of the following components. 1. Antigen, the core component, which is a weakened, inactivated, or a part of the pathogen, such as proteins or sugars from the pathogen's surface. This stimulates the immune system without causing the disease itself. Number 2. Adjuvants. Substances added to boost the immune response by enhancing the body's recognition of the antigen. 3. Preservatives, used to prevent contamination of the vaccine, especially in multi-dose vials. 4. Stabilizers, help maintain the vaccine's effectiveness during storage and transportation. Types of vaccines. 1. Live attenuated vaccines. These contain a weakened version of the living pathogen that cannot cause disease in people with healthy immune systems. Examples include the measles, mumps, and rubella MMR vaccine. Number 2. Inactivated vaccines. These contain killed pathogens or inactivated forms of the virus. An example is the polio vaccine. 3. Subunit, recombinant, polysaccharide, and conjugate vaccines. These use specific pieces of the pathogen, such as its protein or sugar. The COVID-19 vaccines from Pfizer and Moderna are mRNA-based subunit vaccines. 4. Toxoid vaccines. These are used to protect against toxins produced by certain bacteria, such as the tetanus and diphtheria vaccines. 5. mRNA vaccines, a newer type of vaccine that uses messenger RNA to instruct cells to produce a protein that triggers an immune response, such as the spike protein in COVID-19 vaccines. How does vaccines interact with our bodies? When a vaccine is administered, the immune system responds in several ways. First, the recognition of the introduced antigen. Once the vaccine is introduced into the body, the immune system identifies the antigen, the harmless fragment of the pathogen or a weakened pathogen. After recognizing the antigen, the immune system sends cells like macrophages and dendritic cells to fight or capture the antigen, which they then present to T cells and B cells. Soon as B cells are activated, specific antibodies that neutralize the antigen is produced. These antibodies attach to the pathogen and prevent it from infecting healthy cells. After defeating the fake pathogens, some B cells and T cells become memory cells. These cells remain in the body long after the vaccination, allowing the immune system to respond rapidly if it encounters the pathogen again in the future. Because the immune system keeps a record of every disease it eliminates, just in case it encounters it again. Are vaccines really effective? The effectiveness of a vaccine depends on various factors. 1. Herd immunity. When a significant portion of the population, in this case us humans, become immune, either through vaccination or past infection, the spread of the disease is reduced, protecting those who cannot be vaccinated. 2. Booster shots. Some vaccines require multiple doses, booster shots, to maintain immunity. This is because the immune response can decrease over time. 3. Immunogenicity. This refers to the ability of the vaccine to trigger the intended immune response. 4. High immunogenicity, which typically correlates with long-lasting protection. How are vaccines produced? Vaccine production involves several careful and meticulous steps. The first step is research and development. Scientists identify suitable antigens, study pathogen biology, and develop the safest method to deliver the antigen into the body without mistakenly actually killing the person. The next step is preclinical testing. The vaccine is tested on animals with similar DNA as humans to assess its safety and efficacy. Then clinical trials. After passing preclinical tests, vaccines undergo 3 phases of clinical trials in humans to evaluate safety, optimal dosage, and effectiveness. The next stage is regulatory approval. Agencies like the FDA or EMA review clinical data to approve vaccines. If the vaccine is tested and proves not fatal to humans and actually perform the intended function, large-scale production of vaccines is done with growing the antigen, purifying it, and formulating it with other ingredients like adjuvants. Manufacturing must adhere to stringent good manufacturing practices, GMP, to ensure quality and safety. The last stage is distribution. The vaccines are transported under specific conditions to preserve their stability. Some, like mRNA vaccines, require ultra-cold storage. Vaccines undergo rigorous testing to ensure safety. Once licensed, they are continually monitored for side effects through systems like the Vaccine Adverse Event Reporting System, VAERS. Side effects are usually mild, such as swelling at the injection site, while serious side effects are extremely rare. Vaccines not only protect individuals, but also communities. The concept of herd immunity means that when a significant percentage of the population is vaccinated, the spread of contagious diseases is greatly reduced. This protects those who cannot be vaccinated, such as people with weakened immune systems or allergies to vaccine components. Vaccines represent one of the most powerful tools in modern medicine, preventing millions of deaths each year by priming the immune system to recognize and combat dangerous pathogens. Through rigorous research, development, and safety monitoring, vaccines continue to evolve, ensuring protection against an ever-growing list of diseases. Follow my page to learn more everyday knowledge.